A warm welcome to everyone. Today, we will cover the topic genetic code and its characteristics. What is nucleotide base of DNA? In the DNA, there are numerous nucleotides, namely adenine, guanine, cytosine, and thymine. These together form a nucleotide. Now, central dogma. It is a two-step process, namely transcription and translation. In this, transcription is a change of DNA to mRNA and translation is a change of mRNA to polypeptides, which are the functional products of protein. Codons. Ribosomes decodes mRNAs by reading their nucleotide sequences in groups of three, namely codon or triplet codon. Letter A, C, G and T corresponds to the nucleotides found in DNA, and A, U, G and C corresponds to the nucleotides found in the RNA, which are organized into codons. The genetic code consists of 64 triplets of nucleotides. Each codon encodes for one of the 20 amino acids used in the synthesis of protein. The collection of codons is called genetic code. This determines a protein amino acid sequence. 3 nucleotides in a row counts as a triplet and code for a single amino acid. Therefore, each sequence of 3 codes for an amino acid. Each codon is a triplet nucleotide, that is 64 codons, out of which 3 are stop codons, therefore 61 codons for 20 amino acid. In the side given is a table showing the amino acids with its coding codons. These codons have many features. First, most codons specify an amino acid. Then, 3 stop codons mark the end of a protein. 1 start codon, AUG, marks the beginning of a protein and also encodes the amino acid methionine. The picture given below exhibits all the features of a codon and clearly shows an amino acid sequence. Now, coming to our main topic, genetic code. It is a set of rules and regulations used by living cells to translate information encoded within genetic materials, that are DNA and RNA. Codes in mRNA are read during translation, beginning with a start codon and ends with a stop codon. They are read from 5 to 3 and specify the order of amino acid in a protein from N-terminus to the C-terminus. The given picture clearly shows the 5 to 3 specific order and a genetic code. The genetic code has multiple characteristics, the first being triplet nature. A triplet code could make a genetic code for 64 different combinations and provide plenty of information in the DNA molecule to specify the placement of all 20 amino acids. Then the next being specificity of genetic code. A specific codon always codes for the same amino acid. Universality of genetic code. In all living organisms, genetic code is the same except for mitochondrial codon. AGA and AGG code for arginine in cytoplasm, but in mitochondria, they are a termination codon. Degeneracy. The code degenerates, which means that the same amino acid is coded by more than one base triplet. Example, three amino acids, arginine, alanine, and leucine, each have six synonymous codons. No overlapping of genetic codes. All codes are independent sets of three bases. Redundancy of genetic code. Although each codon represents to a single amino acid, a single amino acid can have multiple codons. Commaless. There is no signal to indicate the end of one codon and beginning of the next. Thus, it is comma-free. Polarity. The genetic code has polarity. The code is always read in a fixed direction 5 to 3. AUG initiation. It is initiator codon in majority of protein. Methionine is the only amino acid specified for one codon, AUG. 3 stop codons. There are 3 codons out of 64 in genetic code which do not encode for any amino acid called stop codon, UAA, UAG, UGA. Now, let's entertain ourselves with some facts about genetic code. Genetic code was first discovered by Nirenberg and Massey. The genetic code necessary for building up its own amino acids during the multiplication of virus is carried by the virus nucleic acid. The simplest amino acid is glycine. Mutation is due to the changes in the genetic code. Humans share 99.9% of their DNA. Har Gobind Khorana is the interpreter of genetic code. Now these are some questions given along with their answers regarding genetic code. Define genetic code. It is a set of rules and regulations used by living cells to translate information encoded with genetic materials, DNA and RNA, which is or are the initiation and stop codon. AUG, most common initiator, UAA, UAG, UGA, stop codon. Genetic code consists of how many codons? 64 codons each with 3 nucleotides. What are the characteristics of genetic code? Triplet nature, non-ambiguity, universality, degeneracy, no overlapping, redundancy, commaless, polarity, AUG initiation, and 3 stop codons.